Thank you, everybody. Good afternoon. This is my first time in China. I want to thank the organizer for this wonderful opportunity to visit this amazing country and be part of this incredible community. I am a neuroscientist, so we will take a step back from the last talk and focus on preclinical research. That is the research that then brings us to the development of drugs for the brain. So my interest is to try to understand what happens to the brain when we lose the ability to be who we are as a consequence of life events. And the most common that is inevitable for everybody is obviously aging as well. And as we have heard a lot about this importance of health span, we have not talked much about what is the consequence of increasing actually also lifespan for the brain. Because as we increase lifespan, we increase the risk for dementia. And so we need to take an active stance from the preclinical research to develop something for the brain so that we can age well, not only on the skin, on the body, on the joint, on the heart, but on the brain because it controls everything. I was a TBI investigator, a professor for almost 20 years before joining Altos, and I will tell you about Altos. And we have been working for many years in trying to understand how the brain loses their functions and how to restore it. And we have used different model system with different disease system that spans from aging to neurodegeneration, traumatic brain injury, which is what I will talk for the next 20 minutes, and but also brain radiotherapy, which is used to treat cancer and induce persistent and permanent loss of cognitive function that affect quality of life. My group has also been working with NASA for many years, for 15 years, and in Pioneer, the studies of the brain on space, as our astronauts want to go into space. But as we know from medicine in space, we also learn a lot from what is going on in the Earth. And so we have worked on many different pathways that we have studied. And I want to make a quick link before I go to the talk on the what actually Steve said earlier, that if you want to age well, you should be a woman, right? So our work with NASA actually was the first to demonstrate that the female mice are in fact protected from deep space radiation. And this came out during the time where there was the whole discussion, in fact, for having a crew only of a female to go to space, to Mars. So being a female confer a lot of resilience and also confer the possibility, if you want, to also go to space perhaps and being more resilient than men. So I moved to Altos Labs 3.5 years ago from my academic position. And our mission is to restore cell health and resilience through cell rejuvenation and to reverse disease that are and disability that occur as a consequence of life. In my specific, we focus on the brain. And our approach to restore brain health is an holistic approach that has to start from understanding within the cell, from the nucleus and the cytoplasm, how the cell works. You can't see the cogs, there are cogs there, into systems, because the brain is extremely difficult tissue that is not as many cell types that have to interact together. And so that we can study, and from the understanding of the cell, we can study the whole system. And so we can then bring it to the human and find the interventions. So we have established in vitro and in vivo physiological mechanism, and we can test intervention to restore brain health. So now at Altos, we are focused primarily on aging and neurodegeneration, as well as traumatic brain injury. And so I thought that I wanted to give a talk that will expand perhaps your understanding, as you have heard a lot about neurodegeneration, of another environmental risk factor, which is actually the strongest environmental risk factor for developing dementia, which is traumatic brain injury. And Traumatic brain injury is very wide. There are over almost 30 million of people that suffer of it. And this is as a consequence of a motor vehicle accident, but the most common is really fall in our everyday activity. And especially if you are over the age of 65, is extremely common and is deleterious because it increases the risk of dementia. So you may be familiar with this graph. Here we see in the y-axis age, and we know that as you age, there is a cumulative risk of developing dementia that increases, especially after the age of 65, increase very sharply. But this curve, while we want to push it to far to the right, this curve can be pushed way far to the left after trauma, after traumatic brain injury. And in fact, in blue you have what happens if you have a severe trauma, so the risk of dementia is extremely increased. We see that even with a mild concussive injury, there is over twofold of increase of developing dementia. And so we wanted to study this both from the human perspective to understand what happens, but also recapitulate in mice models that can help us to address the problem of aging, because it is a form of accelerated aging. We have several different mouse models, and I'm showing you one here. It is the control cortical impact, where you can see, maybe I can the laser doesn't work, but that's okay. You can see on the top row in A, MRI of animal that receive a contusion injury, and over time they develop this cavitation, which is similar to what we can observe also in humans years or months and years after traumatic brain injury. So we can observe those gray areas on the and we can recapitulate several other aspects of what we see in humans. For instance, there is the release of a chemokine with a small protein from the brain that is also a biomarker that is released after trauma and is found in the plasma and is found both in humans and in mice. And in fact, it can be used as a biomarker. And this, in fact, is called CCL2, which is also a senescence marker. And this is highly elevated in the mice up to like very early on, 7 days after trauma, and remains elevated for a long time, further demonstrating that there is an accelerated aging. So giving the trauma to the mice, we can exacerbate this aging phenotype in just 1 month, where we can measure cognitive deficits, we can measure inflammation, and we can measure neuronal dysfunction. So this is just an example to show you how we measure loss of cognitive functions in mice. There is many ways. This is one that really resembles how you navigate every day on your world. You navigate and you live your life based on maps that you form around you. So you know where the grocery store is, you know where home is, you know where work is, based on cues that are around you. When today we came here, for some of us was the first day, so we had to navigate a little bit to learn where the conference center is compared to other cues, so that tomorrow actually I will not make any mistake, I will come here straight. We can measure those same functions in the mice, and those are in fact affected during aging, and they are equally affected after traumatic brain injury. So we can test the conference center, the ability of the mice to find the conference center just by putting them in a pool with opaque water, and they don't know where the conference center is. Then we show them one time, and then they learn quickly. But if they receive trauma 1 month, 7 months, 8 months before, or even 30 days, they are impaired to the same level as old mice. And as I said, there is a strong inflammatory component, which is characterized by those inflammatory cells, the innate immune system of the brain, which are the microglia. And if we zoom in, we can see in red those microglial cells that are part of the inflammaging process that affect the neuronal function. We have previously shown their interaction with the neurons. And as you can see, you can appreciate, if you compare those two images, that also in the old mice brain, you find those similar cells, and also in the Alzheimer's disease mice brain. So this mouse model of a traumatic brain injury recapitulate the human phenotype. And phenocop is also age and neurodegenerative neurodegeneration, but in a much shorter time frame, because there is memory deficits that occur as early as 7 days and they last persistently. There is a loss of neuronal connections that are those that are essential to form maps and memories. There is this chronic inflammatory process, and there are epigenetic changes which I didn't show. There is also the methylation clocks, of course, that we have done with Steve, and they are also affected to the same level that we see in the aging brain. And there are senescence markers. So we aim really to understand the root cause of those changes so that we can revert those deficits. So my group has worked extensively to try to understand those mechanisms. And here is just a cartoon to the left of a simplified cartoon of the brain where there are the neurons, microglial cell, astrocytes. They all release all those molecules that can induce activation of each other cells. They can go in the circulation and, in fact, call in peripheral cells to enter in the brain. Specifically, I mentioned earlier the CCL2, which is a chemokine, small protein that is released after trauma. It is also found in the aging brain and is a signaling molecule that goes outside in the periphery and serves as a signaling for macrophages that from the bone marrow, they enter in the brain by chemotaxis. So we have previously shown that actually if we can affect the different cells, we can in fact revert the loss of cognitive function. One of those was microglia. We have previously shown that we can rejuvenate a microglia, and rejuvenation not by transcription factor, and we let a new microglia come in. Those are just like embryonic microglial cell, and we've shown that they can change the environment, and then the neurons start to work properly, and the brain function well. We have shown this with traumatic brain injury, with aging, and also with space radiation, so it's a common mechanism. But then what I want to focus on the next few minutes is, so working on the cells inside of the brain, as you can imagine, is difficult, right? Because of the location, because of where is it in the space. So if we can affect peripheral cells that then enter in the brain, it's a much better vehicle system to get into the brain. So because we have shown, in fact, that microglia release factor that then calls in macrophages, a number of years ago, we tried actually to modify the macrophages. And we saw that if we can prevent the infiltration of the macrophages that acutely invade the brain after trauma, and actually they also enter during aging and neurodegeneration. If we can prevent the infiltration of those macrophages, we can restore or prevent the inflammatory milieu that then is conducive of cognitive loss. And so we have seen that. So a step forward, though, that we wanted to ask was, what do those cells do when they enter in the brain? We know that if we prevent their infiltration, is good, because even the microglia stop to have that inflammatory state, the brain works well. So what do? So we wanted to ask, because those are peripherally derived bone marrow cells, and they are distinct from resident microglial cells. So we use a fate mapping system where we could precisely and temporally label those cells once they enter in the brain, and persistently and permanently. And we actually saw, to our surprise, that they enter, as we already knew, but they also persist in the brain for a long time, up to 8 months after injury. And here, those are cross-section, coronal section at the level of the brain, where in red we can label those cells, but they don't come out too nicely on this image. We also look, what do they do? So they are distinct from microglial cell in their origins. Microglial cells are yolk sac derived, they infiltrate the brain earlier on during development, and they remain in the brain. Macrophages are bone marrow derived, so very distinct. And they enter, in this case, the brain as a result of an injury, as a result of disease, or as a result of aging. And once they enter in the brain, we have shown that they start to express a myeloid cell marker, which is called IB1. It's a general marker, so by 8 months, actually, they all express this marker. But when we look for markers of microglia, we see very few expression of microglia. So those cells, they remain, though they are in the myeloid system, they remain distinct from microglial cell all through the 8 months that they are there. We also did a transcriptomic signature, and so that still at 8 months after infiltration, they have a persistent signature that is distinct from the signature of microglia. And in fact, this signature is extremely different from microglia because they show that there is elevated marker of aging, of neurodegeneration, and in fact also elevated expression of senescence such as P16 and P21 compared to the microglia that is in the injured brain that is causing inflammation. So they are exacerbated compared to the microglia that is already elevated. And so we took all this data from different animal models and we put together with data that was also published in the literature to find a common signature that is selectively not of myeloid cells, but of peripherally derived macrophages that engraft the brain and persist there. And we found actually 114 unique genes that are only expressed on those macrophage-derived, and they actually have a specific expression of the immune response. They are associated with the immune response. And very importantly, 31 of those core genes, of those 114 core genes, can be found in the druggable genome. Means that there are drugs that can target, that are approved for human, and that can encode for protein that are capable of binding to those molecules and they can be used. So this is extremely important because previously we used only one compound and now we know that we have more. So there is this shared transcriptomic signature across different models that reinforce this notion that those infiltrating macrophages persist in the brain and can be deleterious. But this is in mice. So we wanted to question, is this also evident in humans? So we did a single nuke RNA sequencing from the frontal cortex of patients that had received a traumatic brain injury in their lifetime, but they did not die of traumatic brain injury. So they had TBI years before. Because remember, TBI is this like shift the curve for neurodegeneration. And we found that when we look for the human orthologs of those genes, there is in fact an increased expression of this signature in the myeloid cells in the patients that receive a traumatic brain injury during their lifetime. We further used the Seattle Alzheimer's disease brain cell atlas, and we plug in this signature and found that this signature is also overexpressed in the myeloid cells of Alzheimer's disease patients that had cognitive decline compared to cognitively intact patients. So to conclude, we've seen that trauma in humans and also in mice induce this infiltration of macrophage-derived cells that are entering in the brain, they persist in the brain for a long time after injury. They retain their function, I didn't show you, but they are not just red dots in the brain, they are still phagocytically active. And they show a transcriptomic signature that is maintained with age, disease, and across species, and across perturbation. So the next step could be to leverage those findings to precisely target the macrophages and promote brain health in injury, but also in disease. And I want to really end on perfect time, actually. I'm proud of myself. I really want to end with you with this concept that there are different ways we can, in fact, take care of the brain. And one of those that I explain is really from the periphery, which perhaps is the simplest way. But there are different ways, and it's important to really consider the brain as an organ as we talk about longevity. Oh, and one example, since I was making the link with space, that we had already seen also from our space study, that the peripherally derived macrophages can serve as a blood biomarkers to predict cognitive decline in the male, because I told you the female, we see that they are spared. Of course, a big thanks to the previous fundings, perfect timing, previous fundings while I was at UCSF, the team at UCSF, my team at Altos, collaborators for this study is actually Steve that is here, and the fantastic team that I have the privilege to work with while I'm here to talk to you and enjoy this company. Thank you very much.
Thanks for the speech. 感谢 Susanna Rosi 教授的分享。观众朋友们有什么问题想要与教授探讨的吗？
Yeah, Susanna, fantastic. There's a lot of coming work on mitochondrial dynamics and interchange between cells, particularly in the context of immune cells. Did you look at all to see did you stain for mitochondria? Did you look at all to see changes in mitochondria and on mitochondrial dynamics in these cells in TBI?
That's a very good question. That's the first question that Jody Nonnari, when we started at Altos, kept asking me, and we've still not gone to the root of that. The mitochondria, in fact, may be the source of everything, right? We go back and forth, but the short answer, we have not looked at the mitochondria on this.
再次感谢 Susanna Rosi 教授。